All right, so for part one of this crystal violet kinetics lab, we need to make a calibration curve for a bunch of various crystal violet solutions, so 4 to be exact. So how you're going to make these is by pipetting deionized water as well as crystal violet together to make solutions of various concentrations. So I have this pipette, this 10 milliliter graduated pipette that I've rinsed with water a few times, and now I'm going to use the pipette increasing amounts of water into 3 clean dry beakers. Right, so I think your experiment has you get 2 mils of water, 8 mils of water, and then I think 6. But so I'm doing that here. I'm pulling up these this deionized water, getting it to about the level I need it to be. Again, your experiment says to get like 2.00 milliliters of water. You do not have to get exactly 2 mils. If you get somewhere close, that's fine. But what is extremely important is that you actually record what you're measuring. Right? So I'm dispensing, tapping off. I would record that measurement and then I dispense it into this beaker. Right? I sort of went a little bit out of order for the experiment. I wanted to pipette my water before I use the pipette for anything else. So now I'm going to use it for the stock crystal violet to make a 25 milliliter solution of it in a volumetric flask. So this is the stock crystal violet from the fume hood. I'm pouring some of it out into a different clean beaker, and I'm going to use this small amount to rinse and condition this pipette. So that way the walls of it are coated with the crystal violet rather than the water that I had just used. So I'm not diluting, unintentionally diluting the crystal violet solution. Okay. All of the data for this lab that you collect are going to be absorbance values, and we need those to be concentrations in order to do the actual kinetics part of the experiment and figure out what the rate law is for this crystal violet hydroxide reaction. So it's extremely important that you get the concentrations of these solutions right, because you'll be using these solutions concentrations to make the calibration curve. So be very careful to not sort of cross-contaminate your solutions when you're doing all of this. So your experiment says to get 4 milliliters of crystal violet. Again, it does not have to be exactly 4.00. I got somewhere close. I would record what I used, and then I would dispense it into this volumetric flask, and then I'm filling up to the mark with deionized water. Right. So I have bent over, and I am staring at the white line in the neck at eye level, and I'm just squeezing the squirt bottle until I reach that line. Be very careful with this. If you do overfill, you have to just start over in making this crystal violet solution. So I've got it. I'll put a cap on it. And while holding the cap, I'm going to invert this thing 15, 20 times to mix it all extremely thoroughly before moving on. Right? So again, for the calibration curve part, your experiment does say to use very specific volumes of crystal violet and water. As long as you're close to those volumes and you actually record what you've dispensed, that's totally fine. So for example, if it called for 8 milliliters of crystal violet and you got like 7.91, that's close enough, but just record what you actually have. Right? So now I'm transferring all of that into a new beaker. Now I have to condition another pipette. So I'm pouring out some of this solution that I just made into another clean dry beaker to then coat the walls of this thing with that solution. So again, all of the data for the lab depends on you making these first 4 solutions really, really well. Right? So just be very careful to not cross-contaminate things. Record your actual measurements, and you should be good. Right? Record your actual volumes measurements. Don't. So again, just conditioning this pipette, rinsing it with the solution I'm going to put into it, emptying that out. All right. So now I'm going to pull up, I think, 8 milliliters of crystal violet. Again, I'm not going to bother getting exactly 8, but I'm going to get somewhere close and record what I actually want. Right? So that stock crystal violet just moved off to the side so I didn't accidentally like grab it and try to use it. So this is this crystal violet that I'm pipetting from, if you've lost track, is the crystal violet that I made in the volumetric flask. So I'm getting to about 8. I would record this volume. Once I get there, tap off any drops that I have, and then let it go into one of my beakers. Right. Always tapping off the tips of the pipettes because that has been dispensed, so you need it into the container. Again, this other one. Again, I'm sort of leaning over, trying to stare at the graduations at eye level. Once I think I get it close, tap it off in the container, record that volume, and then dispense. So in this video, I don't record the volumes because I was trying to sort of just show you how to do the experiment. But if I were actually collecting the data, I would be recording all of these volumes to the hundredths place. All right, so I now have made my 4 crystal violet solutions for the calibration curve. I'm going to give them a little swirl to make sure they're thoroughly mixed. And then we're going to take these 4 and we're going to shove them into our colorimeter to measure their absorbances at 610 nanometers. Right. So sort of moving this stuff out of the way really quick. So it's been a while since we used these, so I'll go over it. There's a green button on it, and that's how you actually blank the thing. So before we put any of these samples in, I have to zero it with just deionized water. I'm grabbing a piece of chem wipe, a tissue paper here, because my fingerprints will get stuck on the glass, and those can potentially absorb light, so we just want to make sure they're not on there. So fill this with deionized water, wipe off my fingerprints, put the lid back on, put it into the chamber, close the lid, and push the green button to blank it. This takes time, probably 10-15 seconds. There's a little green indicator light on that green button, which is a very bad design, but once that green light turns off, it should zero, and you should read a 0 on your calorimeter, or on your Pasco data logger. Now you'll notice it said 4.309 times 10 to the negative 4th. That's 0.0004. Right, so that's effectively 0 because the measurements on the Pasco really only go to 3 decimal places anyway. So I dumped the water out, and now I'm rinsing and conditioning the vial with the first solution. So put it in, shake it up, dump it out. Right, do that again. Right. I want to make sure that as I'm transitioning from one solution to the next, I'm not accidentally diluting the next solution, right? So rinse it out twice with the solution I want to put in it, fill it up, cool, close the lid, wipe my fingerprints off the thing, put it in, and it will display an absorbance reading at 610 nanometers, which is 0.190, so I would record that number, right, for the first solution. Now we're going to continue to do it for the rest of them. Right? So just throw that out into a waste beaker, rinse with a little bit of the second solution, give it a shake, and we're going to repeat this process until we go through all 4 of them. So always rinsing a couple of times with the new solution, and then filling it, shoving it in the instrument, recording the absorbance. Right? This seems really sort of painstaking, but it is extremely important to get good data in the actual reactions. We need to have the relationship between absorbance and concentration for crystal violet down, right? Because in the reaction parts, right? So there's the second one, the absorbance went up, and when we're reacting crystal violet and hydroxide, how you're actually watching that reaction proceed and monitoring the rate of the reaction is through the change in the absorbance of the solution, because you're taking crystal violet, which is purple, and reacting it with sodium hydroxide, which is colorless, and the product is colorless. So our absorbance is going to go down over time, and that's how we're measuring the rate. So those absorbance values need to have concentrations that are known to go with them. So Being very careful to not mess up these solutions is key. I'm almost there. I think I've got one more solution to do after this, right? Right, so there's our new absorbance for the third one, 0.654. Dump this whole thing out, and then rinse with the final solution and fill it. So I think I only do one rinse with this. Always be mindful about how much solution you have left. I didn't think I had enough to do multiple rinses, so I'm just doing one, and then I fill with the remainder. So, gonna put this in, get our final absorbance value, which I think is around 0.9, if I remember correctly. Close this lid. I think it's about 0.9. Yeah, okay, 0.974. Right. So that's the first part. That's making the calibration curve. All of those solutions we can just get rid of, which have been added to the waste as we've been going. Right. And so for setting up part 2, I need to make 2 25 milliliter volumetric flasks. So I'm getting all this part 1 stuff out of my way, and I'm making 2 volumetric flasks. One that will contain sodium hydroxide, and one that will contain crystal violet. So this is the same one from earlier that I used to make the crystal violet. So I'm just rinsing it out a few times to make sure that the walls of it are just coated with deionized water rather than the crystal violet solution that I made up. Right. So giving it three, maybe 4 good rinses. Right. Same thing with my little cuvette. I just want to get that stuff out of there. All right. So 2 volumetric flasks, one for crystal violet, one for sodium hydroxide. So this is the stock crystal violet again that's from the fume hood. This is not the stuff that I made up into a volumetric flask in part 1. It's just straight from the fume hood. Right? So again, I'm pouring out a portion of that to rinse and condition this pipette. And once I do that, I'm gonna then, I'm gonna fill it to, I believe your experiment says to get exactly 5 milliliters of crystal violet. This one you should strive to get exactly 5 for reasons that will become apparent when you go to do the report. You're going to run 2 reactions. From the first to the second reaction, you're going to change the hydroxide concentration, but keep the crystal violet concentration the same. So you should aim to get 5 milliliters of crystal violet both times when you do this. Right, so I'm sort of low on crystal violet, and I don't want to go over to the fume hood to get more, so I'm tipping my burette, holding everything, and getting 5 milliliters. So again, lowering myself down to get to eye level with the 5 milliliter mark, lowering a bit, tapping it off. Record that volume, which for this should be almost exactly 5, and dispensing that into a clean volumetric flask. All right. I'm going to grab another pipette. This one is dirty from earlier. It's still got crystal violet in it, so that's okay. I'm going to rinse it out with the sodium hydroxide solution. Right. So I didn't have a sink to dump this into. That's just deionized water, so I'm going to chuck that out. So I have a beaker that I can put the sodium hydroxide into. So I'm gonna pull up some of the sodium hydroxide again, coat the walls of this pipette. I'm rinsing out the crystal violet that's in there before I do this, but regardless, I still have to condition. I still have to rinse this pipette with the sodium hydroxide solution I want to put into it. So I'm gonna pull up some of it, I think 4 or 5 milliliters worth. Then I'm gonna take it out, rinse the walls down as I have been throughout this entire video. Right. And then get rid of it. I'm going to do that one more time. So conditioning twice with the sodium hydroxide solution. And it's not much, it's just like 2 mils or so, and then I'm just coating the walls, 2 milliliters, empty the thing out. And now I'm ready to draw up my sodium hydroxide solution. Pull up the volume above the volume I need, pull it out of the liquid, drain until I get to the mark that I want, and then drain it into this volumetric flask. Tapping it off. Right. So I've got these two flasks. I have the sodium hydroxide I need in them, I have the crystal violet I have in the other one, and I need to fill them both up to the mark with deionized water. Both of these are going to go in that cuvette eventually. Right, so staring at that white line at eye level, trying to pull them closer to me so that's easier to do. So I'm only, it looks further, but I think I'm only like 4 inches away from these flasks at this point, where I'm staring at them with my eye. So get it to the mark. All right. Done. Do the same with the crystal violet one. And again, if you are not comfortable with filling these with your squirt bottle, use a dropper pipette. Right? Go slow when you get in the neck. Make sure you don't overfill. If you do overfill, you have to restart. So just be careful and methodical. Right. Caps on both. And then invert them all, both of them, I don't know, 15, 20 times, somewhere like that. Right? So this takes a bit. So once I have these thoroughly mixed, I'm going to put each of them in their own beaker. Clean and dry beaker. Doesn't really matter the size. We're just going to pour these things together. So as long as the beakers will hold 50 milliliters, because 25 and 25, right, you're good. It could be, right, so I mean, 2 completely different size beakers, doesn't matter. Right. I'm going to dump the sodium hydroxide into one and the crystal violet into the other. All right. So my 2 solutions are ready to go, but before I dump them together, I need to set up the spectrophotometer, or sorry, the Pasco calorimeter to record the data over intervals of time. So how we're going to do this, sorry for the shaky camera, but we got to go to the home screen, and then we want to go to sensors, which is F4. And it's currently collecting 1 sample a second. That's hard to see, but this sample rate, I'm going to change it to be 30 seconds. So it will collect a data point every 30 seconds, right? Because you're going to run this reaction over like 15 minutes. So if we collected a sample every second, that's going to be a lot of data, right? So I have changed the sample rate. Again, once you're on the home screen, it's just F4 and then change it to collect every 30 seconds rather than every second or whatever the default is, right? So this is all ready to go. I have my cuvette ready. I'm going to pour these 2 together, and then I'm going to give them a quick swirl. And I'm trying to go fast with this, so give it a swirl. Rinse this cuvette with the solution I'm going to put into it. Dump that out super quick. Fill it up. Put the cap on. Wipe my grimy fingerprints off, and shove it in the instrument. And then to start recording data, just like the thermodynamics lab, you push play. So close the lid, push play, and then sit back and relax for 15 minutes. Right? So it doesn't look like anything's happening here at the start, and it's because I set it up to collect every 30 seconds. So there's a data point, and then we got to wait a while. So there's our sort of first data point. For the sake of this video, I told it to collect data every 10 seconds, so we didn't have to wait as long. But when you do it in lab, it'll be 30 seconds. Right? So we just kind of sit and wait until the time passes and the reaction runs to completion. When you're done, you should pull the cuvette out and you should have a solution that's almost colorless, if not colorless, right? Because we can see the y-axis on this graph is the absorbance at the orange wavelength. It's decreasing over time because our colored crystal violet is getting converted into a colorless product as it binds with the hydroxide, right? So yeah, just a lot of waiting at this point. But that being said, you could spend the time cleaning up and prepping we're going to run the second experiment. So use the time wisely in lab, get the volumetric flask prepped for the second reaction as this first one runs, right? So show you guys what the data sort of looks like as we're collecting it, right? So after, I don't know, what, 80 or so seconds here of transcribed. So, but yeah, there's about 15 minutes of downtime here, so use it to prep the next reaction. So good luck.